So thank you for welcoming here. And I just want to start my talk. This is going to be a bit more technical than some of the others. This is focusing on an aspect of the research that we're doing at Domainex. But first, the slides aren't coming up. Yes, they are. But first, a brief introduction to Domainex. We are a contract research organisation based here in the south of Cambridge. We're about 100 scientists, based both between the chemistry building that's at Chesterford Research Park, about 5 miles down the road, as well as our biology building, which is literally just next door. And if you hang around afterwards, we'll be able to give you some lab tours for anybody who is interested in seeing those. We've got a global client base, so a lot of our clients are based in Cambridge, but we also have a lot of clients who are US, as well as Europe, and also in the APAC. In terms of the services that we offer, we have a full integrated service all the way covering from initial target selection all the way through to preclinical candidate nomination. For this talk, I'm going to talk about some of the protein aspects that we do. We've been working with Protein Sciences Group for nearly around about the last 25 years, where we produce high quality reagents for antigens for antibody generations, as well as reagents for assay builds and structural biology. Specifically, this is about an aspect for where we've been working on, I think, focused for about the last 5, maybe 10 years, specifically looking at membrane proteins and producing membrane proteins. So membrane proteins are a really important class of proteins. They account for around about 30% of the human genome, but actually around about half of drug targets are membrane proteins. And this increases once you're starting to take into account therapeutic antibodies as well as other biologics. However, specifically when it comes to working with membranes for isolating proteins, they are challenging to work with. Membrane proteins traditionally have very, very low expression yields when you're trying to make and purify them. And critically, they require the lipid environment naturally to be able to provide them with both the structure as well as the function. And therefore, when you try to strip away that native lipid environment, you can affect both the structure of the membrane protein as well as the incorrect folding and the structure. And if you're basing your whole drug discovery program on that integrity of that membrane protein, you can start off in the wrong direction. Detergents have traditionally been used to isolate membrane proteins. However, there's no universal detergent that people can use, and therefore you need quite a lot of time to validate, make sure that when you strip away that native lipid membrane, that you've still got the correctly folded protein and the function is still relevant. In more recent years, amphimatic copolymers such as SMA have been developed, initially by Tim Defon down in Birmingham. And these have offered an alternative method for producing and isolating the membrane proteins. What these do is they form a disc in the plasma membrane, and then they isolate a section of the membrane out alongside the membrane protein. What this means is you maintain the lipid environment that you've got, which means that the conformation of that membrane protein is more likely to be relevant. However, there are issues, as you can imagine, with this isolation. Early amphimatic polymers have an awful lot of issues in terms of downstream handling, and even now we've got a lot more that have come through onto the marketplace, but they still have challenges in terms of instability and particularly in terms of aggregation. And this can make it incredibly difficult to work with them for downstream applications such as biophysics and especially structural biology. As I said, we've been working on this for a number of years now, and we've got some advances that we want to talk about. And to do this, we want to use a specific ion channel, TRPML3, as a case study. TRPML3 is a calcium channel that's expressed on the membranes of endosomes and lysosomes within the cells. It's a heteromeric calcium channel, so therefore it's got 4 repeating subunits within it. And it's been linked with neurodegenerative diseases such as cancer as well as cell survival. In terms of this case study, initially what we wanted to do was repeat the literature reproduction that's out there in terms of detergent expression and purification of the protein. So we expressed full-length TRPML3 with an N-terminal his tag and a flag tag in XP29 cells as a mammalian expression system. Following literature protocols, if I get the Following literature protocols, we were able to get really good expression levels and a really nice purification system that worked through. When we looked at the size exclusion trace, you can see there's about 40% of the TRPML3 is expressed as a heterotryme, as a heteromeric ion channel. And then here you've got a large number of aggregated proteins that's come through. But once you separate out the aggregates, we're able to get a nice monodispersed protein that's suitable for structural biology. Using the microscope down in Cambridge, we're able to get a really nice 3D cryomaps of the TRPML3 channel, and we're able to get both 2.7 and 3 angstrom maps of this for the agonist bound as well as the apo form of the protein. And you can see on the left, we've got the apo form of the protein. So this is an inactive conformation. And you can see, hopefully on the image, you've got a closed conformation where there's no channel formed. And on the right-hand side, with the agonist, you've got the open conformation. What's particularly nice with this structure is that because we're able to get such a high resolution, you're actually able to see the agonist bound into the channel. And at 2.7 angstroms, this is really suitable to help us enable to do structure-enabled drug design on this target. Based next door, we've also got a really experienced biophysics team. And so what we wanted to do was to develop some biophysical assays to demonstrate actual target engagement as well as the on-off rates that you need to do in terms of drug discovery. And so we immobilized the TRPML3 onto a chip surface and then flowed over the agonist. And what this enabled us to do is obtain binding affinity measurements with the KDs here, as well as the kinetics. And whilst we're able to obtain really nice data, it is worth flagging that the affinities for these compounds in the literature are actually a little bit offset in terms of what you actually get in terms of these cell-based assays these are reporting. What we wanted to do next was compare the detergent purification and the detergent structure we got with one made with these amphomatic polymers. And so we embarked on expressing and purifying TRPML3 in these amphomatic polymers. And we've got a really nice, as I said at the beginning, recently you've had a lot more polymers coming out into the marketplace. And we've got a 96 well plate solubilization screen using all the range of commercially available polymers. What we found with experience is just because your polymer is able to solubilize your protein, it doesn't mean it's in a native conformation that you can extract, and also the affinity tag can be included. What we also run, as well as the solubilization screen, we then look for the Western blot in terms of the membrane fractions, and you can see with all these polymers, there's only a handful of them that are coming through. In this case, you can see the cycloiase, SMA, and astes have all solubilized it. However, once you put these into the extracted proteins, the affinity resin, it doesn't actually mean that they're able to come through. What we also do is once we've managed to extract the protein, we then run a native page gel. And what this does is make sure that any of the isolated protein isn't aggregated, because the aggregates won't run in through the gel. So initially, when we're able to extract these, we're able to get non-aggregated protein moving through into the gel that's suitable for downstream. Moving on from here, we're then able to put these through a size exclusion chromatography. And you can see from the sec trace, you've got oligomeric protein coming through, but you've still got a really nice TRPML3 tetrameric peak coming on. However, this is where problems occur. You get about 50% of protein loss once we take the protein through. And then once this is further concentrated, we still get even more loss. So with this, which is common to most proteins that we've worked with a lot of these polymers, the protein aggregates and it drops out of solution as long as you try to use it. For structural biology, we're not able to concentrate this protein up in this form. It's a high enough concentration to move through into structural biology. However, we have identified a solution which works for a large number of these channels. It's a novel nanodisk solution which stabilizes the nanodisk conformation, which we're calling PureDisk. It improves the solubilization conditions, means that more polymers actually work in terms of solubilizing the protein. It reduces the aggregation that you see widely in polymer nanodiscs, and it enables us to actually concentrate these proteins up to a higher concentration, which helps us with the downstream applications. In terms of the solubilization, you can see on the left-hand side, this is the native page gel that we've run from the standard polymer purifications. And you can see with the cubicles how few bands we're able to get, so how few of these cubicle polymers are actually able to solubilize TRPML3. Whereas on the right, with the native page, with the pure disc, you get much more solubilization, and all of these are moving through into the native gel, suggesting that we've got monodisperse protein coming through. One of the best illustrations we've got are the 3 different size exclusion chromatography traces. On the left-hand side, you've got the detergent-produced TRPML3, where you've got a large peak of the aggregates and oligomers relative to the tetrameric protein peak that we're looking to see. In the middle, you've got the size exclusion trace with the standard polymer nanodisc, and on the right-hand side, you can see what we've got coming through with the pure disc, where you've got one single really nice peak of protein. And then when we look through a DLS, for those who aren't aware of DLS, it's looking at monodispersity, specifically of the protein. So on this left axis, you've got the radius of the particles, and you can see with the standard polymer prep, this is the issue that we always see, where you get aggregation as soon as you start to concentrate it up to a level that we would deem usable, especially for structural biology. On the right-hand side, you can see the comparison to the pure disc approach, where you've got a really clear single band coming through for the DLS. We've concentrated this up now to above 0.5 mg per mil. We stopped when it got to 0.7 because this was all we needed, but we're able to concentrate this up for downstream. We then took the protein up to the Titan electron microscope up at Leeds to be able to place this onto grids and to get a cryo-EM map. And you can see we've got a really nice 2.8 angstroms cryo-EM map of TRPML3 for the first time in a polymer nanodisc. Go back one, yeah. One of the highlights of this structure specifically, as I mentioned before, one of the challenges you get with detergent purifications is you strip away the lipids, and that can affect the conformation. With this preparation, you can see the lipid is actually bound in the agonist binding pocket. There's some other interesting differences that we're able to see in terms of the cryo-EM structure with the polymer nanodiscs versus the detergent. There are residues here, H2H3, which is critical for the channel inhibition in low pH, and 361 involved in sodium-mediated channel inhibition. These are resolved in the nanodisc cryo-EM structure, which we just don't see clear resolution for in the detergent purification. Moving through into biophysics, we've got a spectral shift assay that we developed to measure binding affinities of compounds, antibodies to these membrane proteins. This is measuring a wavelength shift with a fluorophore that's bound to the target protein. On the left-hand side, you can see this is the detergent purified protein, where we don't see any compound binding. But on the right, with the pure disc purified protein, we really get good, clear binding affinity curves. And what's particularly interesting is when we compare this to the chip-based method, we actually get much more data that's much more in line with the literature reproduction and the cellular data, suggesting that this is a much more native conformation for the protein. It isn't just ion channels, it isn't just TRPML3 that this works with. This is CB1, the GPCR. And you can see at the top, this is a traditional nanodisc purification for CB1, where you're getting only handfuls of amphibatic polymers that are able to purify the protein, and compared to the pure disc purification, where, to be honest, in this instance, almost all of the polymers we tried were able to solubilize the protein. And again, with the CB1, once we try to concentrate it up, here at 0.2 mg per mil, you're getting a large amount of aggregation that you can see. Whereas with the pure disc, it's going up to 0.6 mg per mil, you're getting another nice monodispersed population. So we think this is actually quite a game changer in terms of membrane protein isolation. It means that we can get native conformations of protein coming through that was suitable for antigen generation, biophysics, as well as structural biology, which can be critical for antibody generation, antibodies looking at the structures, as well as small molecule-based drug design. So please come and talk to us. As I said, we'll be hanging around. If anybody wants a lab tour, then you can come and have a chat about this technology or any of the others that we offer. I just want to acknowledge everybody in the department and everybody who's been working on the project over the last number of years. Thank you.